So a few words of introduction about Edgar Walker. So I actually discovered Edgar as the first author of a very nice bioRxiv paper that was eventually published in Nature Neuroscience. And then I rediscovered him as part of a smaller writing group that Uejima put together following my visit at his lab at the end of 2019. And that's because Edgar did his PhD under the joint supervision of Andreas Tolias at the Baylor College of Medicine in Houston, Texas. And as a reminder, we had invited Andreas 3 years ago at this very seminar. So joint supervision by Andreas Tolias and Uejima at New York University. Now Edgar is a postdoc working in the lab of Fabian Zins at the University of Tübingen in Germany. And Edgar is interested in the notion of a neural code at the level of populations of neurons, in particular sensory representations that are associated with uncertainty. And to this end, he leverages the use of artificial neural networks. So that's something that is getting a bit trendy nowadays. In some studies, that's a bit the cherry on the cake. So that's nice, but not very useful. And that's not the approach of Edgar. In Edgar's approach to the study of neural coding in large populations of neurons, artificial neural networks are really indispensable. So today is going to present two applications of artificial neural networks to the study of sensory neuroscience. So thank you very much, Edgar, and the floor is yours.
All right. Well, thank you, Florian, for a very kind introduction. It's always hard to talk whenever someone gives you a very kind and nice introduction, but I'll try my best. So thanks again for this opportunity to present my work. I hope you enjoy it. Please feel free to interrupt with questions in the middle, but perhaps some of the more heavier questions will be better suited towards the end when there's a time for the Q&A. So before I dive into the meat of my talk, I would like to give perhaps a bit more introduction about myself, but really not much more since Florian pretty much gave it away. So again, I'm a computational and theoretical system neuroscientist and machine learning researcher by training, although my background and training for my PhD years really lies in monkey electrophysiology and system neuroscience training, having performed recordings from macaques myself in behavioral training, and then got into a lot of recordings, or at least working with experimentalists to work on large population recordings in mouse, as you'll see some of the work. At the moment, I'm a postdoctoral fellow at Zins Lab in University of Tübingen, particularly furthering my approach of using, as Florian mentioned, machine learning, particularly deep learning approach to understand population of responses, how they encode information from the particularly visual sensory stimulus, and then subsequently utilize this information as the subject involves themselves in behavior and decision-making process. So what really kept on driving me towards all my training has been my passion on a simple question, but as we all know, simple question tends to be the hard questions. So how does the brain arrive at complex decisions and behavior in response to sensory information critically filled with uncertainty? And I think nothing illustrates this point better than seeing the brain actually in action. And in my broad search of internet, nothing illustrated this better than a cat reacting to a piece of cucumber. So let's step back and understand what's going on here. So when cats are presented by the image of cucumber, this hits their retina, climbs up the optic fiber, causes vigorous firing in areas such as primary visual cortex or V1. And then subsequently, this information travels downstream to areas like prefrontal cortex or decision-making area to ultimately arrive at a decision and behavior. In this case, the vigorous jumping you see. So I can already take this and simplify this into this rather simple flow diagram, taking from stimulus S to the response of visual cortex like V1 R to the ultimate decision and behavior B. And I'm obviously very interested in understanding this entire pathway in its fullest detail. And in doing so, I've been taking particularly two complementary approaches to describe this entire system. On one end, I employ a system identification approach using numerical and statistical techniques to characterize the mapping between these observed variables, such as the responses in terms of the stimulus or behavior in terms of the population of responses. And in the past years, we've been seeing a surge of deep learning to model these mappings, and such learned mappings may have been in turn actually be useful in potentially improving the machine learning models such that they can actually take advantage of whatever the particular mapping the brain seems to be using. On the other end is to build what we call the normative theories, which is actually really thinking about how the brain should compute with sensory information to achieve decisions and behaviors. And I'm particularly interested in normative theories of probabilistic computations in the brain, trying to capture brain's computations in terms of probabilistic distributions and all sorts of computations surrounding this framework of probabilistic computations. And whichever approach I take, it's really been fascinating to see a recent explosion of neuroscience dataset, both in size and complexity, ranging anywhere from 2-photon or multi-photon imaging that can achieve now tens of thousands of neurons recorded simultaneously, to advanced in electrophysiology that gives extremely high temporal fidelity from, again, hundreds to now even thousands of neurons across multiple brain areas, all in combination with increasingly sophisticated behavioral tracking and information. And these informations, this increasingly complex and multimodal information, is really bound to advance our understanding and allow us to ask increasingly more complicated questions about how the brain represents and subsequently utilizes sensory information. But the very complexity itself actually calls for novel ways to handle this data, as traditional methods tends to show its limitation dealing with information and data at this scale. And this is precisely where I believe there's a lot of potential for machine learning, particularly recent advances in deep learning, to really come true and help us make better sense of the data and ultimately ask more involved questions utilizing such data sets. So given this, I've been very interested in combining these two approaches of system identification and normative theories of probabilistic computation to probe sensory-based behavior under uncertainty, supported by the development of machine learning-based methods to model and analyze large-scale multimodal data in answering my research questions. In my recent work, which I'll detail today, precisely reflect these two approaches targeting the primary visual cortex, or V1, and importantly, I approach this from two distinct directions that I refer to as encoding and decoding, and making heavy use of the machine learning-based techniques, heavy use of machine learning-based techniques to model and analyze large population responses from V1. And naturally, the rest of my talk today will reflect these two-part structure, and where I describe the encoding work in part 1 of the talk, and then subsequently decoding work on the probabilistic computations in part 2 of my talk. Now, before I finally get to the meat of this, I'd like to highlight that given this is a mixed audience with different interests and backgrounds, there will be some slides that will contain some technical details that could be interesting to some, but I've made it so that they're not essential to understand the talks. So I've indicated these slides that contain such technical details by titles in parentheses and black star on the top right corner. All right, so with that, I'd like to get started with part 1 on encoding. So obtaining an accurate model of how a population of V1 neurons respond to visual stimulus has been a long-standing goal of system neuroscience, particularly under the subject of system identification. And some of the earliest account of system identification dates back to the pioneer work of Huebel and Wiesel, describing V1 neuron responses to parameterized stimulus such as oriented bars. This work has popularized the concept further of the receptive field, describing spatial responses of these neurons, and also gave rise to concepts such as orientation and direction tuning, typically captured by plotting the tuning curves, which mathematically is the expected response of neurons conditioned on the stimulus. More advanced models, such as the linear nonlinear models for simple cells and energy model for complex cells, allows us to extend our predictions to even wider range of stimulus classes, typically going beyond simple oriented stimulus like an oriented bar. But they have been serving critical roles in thinking about these neurons and design numerous studies that has been very informative. They typically offer quite a limited predictive performance when applied on natural scenes, such as natural images. And in fact, building a good predictive model that works well on natural stimulus has been a major challenge in the neural system identification. Fortunately, in the less than really past decade, we have been seeing surge of work in system neuroscience applying deep learning as the next generation of models. The application of deep neural network typically falls under two categories. In a goal-driven approach, the nonlinear features are extracted from deep neural network, which are pre-trained on a task such as image classification. And these are used then in turn to predict responses of neurons by taking features already learned by this network on the particular task. Work from, for example, DiCarlo's group on V4 in area ITs, as well as some of our recent work on Monkey V1, as shown here, have demonstrated that pre-trained networks like VGG, trained on image classifications on natural images, can be used to actually yield good predictive performance on monkey visual neural responses to natural images, significantly outperforming standard models such as the linear nonlinear neuron as shown by LNP models here. Now, in the alternative data-driven approach, a deep neural network is directly trained end-to-end on the population responses to the stimulus. In our recent work, we have trained a recurrent neural networks on mouse V1 responses to natural movies, achieving state-of-the-art predictive performance, significantly improving upon the performance of standard models, again, such as linear nonlinear models across numerous different types of individuals of mice and across different types of movies. So we may successfully consider that we have trained a neural network that can closely approximate a V1 population response. A fear lurks that maybe all we have done is to replace the brain with yet another complex system that is pretty much like a black box without really offering any glimpse into the understanding of how brain actually works. However, I believe that the well-trained neural network is quite a bit much more than a black box. Namely, these neural networks allow us to perform millions of in silico experiments to deeply probe into the model systems, while taking advantage of the fact that these are neural networks and therefore all components can be deeply probed to the heart's content, and therefore it's making accessible to particular scrutinization that is actually not quite possible in real in vivo system. And this in turn can potentially provide us with even experimentally testable predictions that can then be used as the basis of designs for future experiments. So with this mindset, when I wonder, okay, what could we learn about the visual system from the neural network model of V1? Well, I came to realize that much of the advances in understanding of sensory system originates from identifying so-called good stimulus that can drive the neurons well. This was the case in identifying oriented stimulus for V1 by Hugo and Wiesel, as well as in identifying placement within the room as a stimulus for entorhinal cortex and the hippocampus, leading to the discovery of grid cells and place cells. So with this realization, I asked the following simple question. Can we use the trained neural network to predict responses that have been trained to predict responses of V1 to identify stimulus that will strongly drive the target V1 neurons? In other words, can we find the most exciting inputs for the V1 neurons? And thinking through this, we identified 4 steps to be able to achieve this. So in step 1, we need to first obtain responses of mouse visual neurons to as many static images as possible, thereby presenting these natural images and recording from as many neurons as possible from V1 simultaneously. And in step 2, we then use the mouse V1 responses to these natural images as a training data set to train our deep neural network in an end-to-end data-driven manner. Then in step 3, we'll take these trained deep neural network to generate the most exciting inputs for the selected target neurons, taking very advantage of the fact these are neural networks that we can deeply probe and make a lot of manipulations to be able to come to such image prediction. And finally, in step 4, we verify these generated most exciting inputs, or MEIs, that they are indeed the most exciting inputs by presenting them back to the very same neurons that we trained our models to predict, thereby actually closing the loop. And we achieved this in a novel in vivo, silico vivo closed loop experimental paradigm that we call the inception loops. I'm not going to quite go into why this is called inception loops at the moment. If you're very curious, I could talk about it at the very end. Yes, this is a movie reference. So again, the first step of the loop starts by presenting thousands of natural images to a mouse as we record the activities of population of over 3,000 V1 neurons using dense 2-photon imaging. And there's actually quite a bit of data processing and data science that was involved in making such recordings happen. So I would like to give you a bit of a glimpse of where the data set comes from. So the whole data set actually arises from a Microns initiative, which is a $100 million project funded by IARPA in the US as a part of the BRAIN initiative, with the goal of reverse engineering the algorithms of the visual cortex with the goal of improving machine learning. The program has involved 13 PIs over 10 international universities and has yielded recordings of around 100,000 neurons from 1 millimeter by 1 millimeter by 1 millimeter cube cortex of mouse visual cortex, with over 1 billion synapses reconstructed thereby presenting to be the biggest neurophysiological and connectomics dataset available to date. And within this initiative, I've been leading a group of engineers and scientists in developing data pipelines for the tooth fulcrum data acquisition, pre-processing, and analysis, as well as the subsequent deep neural network training and model selection, which all comes back to give the very data set that becomes the basis of the next step, which is the training of the deep neural network. So given this large collection of population responses to natural images, we can take this as a training data set to train our deep neural network in end-to-end fashion. Here I'm just giving a very brief overview. Later on, I'll be more than happy to step into the details of the network if anyone's interested. And this network was, notably, one thing about this network is that it's specifically designed to incorporate simultaneously recorded behavioral information, such as eye movements, as well as the pupil diameter, to improve overall predictive performance of the networks. And we indeed saw that our trained deep neural network achieved state-of-the-art predictive performance in predicting the V1 responses to natural images, better than the standard models of V1, like the linear nonlinear model, here shown by the y-axis being the performance of the deep neural network and x-axis being the performance of the linear nonlinear neurons. So you can see that here individual points are recorded neurons, and they're much better predicted overall by the deep neural networks. And among these, we selected for 150 neurons per closed loop. That becomes the target for the generation of the next step, which is the MEI generation. So those are the ones indicated by the dark black dots. So in generating the most exciting input, or again the MEI, let's start by considering the simple case of a linear nonlinear model, which is actually equivalent to a shallow neural network. So it's no longer deep, but just shallow single layer neural network with no hidden units and simple output monotonic nonlinearity. So if you take any given target neuron that you're interested in, a linear filter that is applied under linear nonlinear model for that neuron is actually equivalent to the input weights from the pixels of the images leading to the target neuron, which is actually by definition the same as the linear receptive field of the model. And under fixed contrast constraint on the images, it turns out that the receptive field is actually the optimal image you can present to achieve the highest activation for the target neurons. So in other words, receptive field is under the very special case of shallow neural network on the linear nonlinear model, receptive field is a special case of the most exciting input. However, as soon as we extend this onto more complex and deeper neural networks, no such simple correspondence of input weights and MEI exists anymore, and requires an entirely different way of arriving at MEI. So here, what we could do instead is to generate MEI is to take, again, target model neuron that you're interested to generate the MEI for, and we start with some image, usually a white noise image, and feed it through the network to gain activation. We then now, you can use the fact that this is a neural network where every step of the way is completely known analytically to actually backtrack and make a small suggestion or a small perturbation to the input image that is predicted to increase the activation of that very target neuron, thereby modifying the input image ever so slightly. We can then feed this again, get the activation, and repeat this procedure over and over until it gets saturated, at which point we have actually arrived at the optimum of the most exciting input. Mathematically speaking, this corresponds to given the input image x and target neuron response model as f of x. This procedure is exactly equivalent to performing gradient ascent on the input image x via the objective function of f of x. And here's the same MEI generation procedure, but here demonstrated as a movie with the graphical depiction of the model neuron's activation surface here over 2 neurons in the right side. So I'd like to emphasize here that this process of MEI generation will actually correspond to thousands, if not millions, of in silico experiments, where each presentation of new slightly perturbed image would have corresponded to a distinct trial. Something that is just practically impossible to perform in the brain due to both time limitations and other effects such as adaptations. So obviously, after all this procedure, we're interested in seeing what MEIs look like. And here are example MEIs from a single closed loop session over the 150 neurons. And when I first plotted this, when I just really first did this and started plotting this, I was quite surprised to see that they all just didn't look like Gabor filters. In some sense, I was trained to learn that the V1 neurons are just bank of Gabor filters. So I was quite convinced that it should look like a whole bunch of differently oriented in size and scale Gabors, but it wasn't quite so. And in fact, there's quite a bit of variety to MEIs. So some are indeed like Gabors, like you might expect, but some are more like checkerboard-like patterns. And some even looks like they might be going for some sort of a corner detection. So now with such a huge variety of almost unexpected diversity of images, it's time to verify them experimentally. And here comes the last step in which we present these MEIs back to the very neurons we model and generate the images for. And we do this by recording from the same neurons across multiple days of two-photon imaging, matching cells using structural information that we acquire at the beginning of each functional recording session. And now comes the result of actually presenting MEIs back to the very neurons. So here, each column corresponds to a specific MEI image, the most exciting input image, and each row corresponds to a specific neuron, such that wherever they intersect, the color of the block corresponds to activation of neuron X to the MEI Y. And it's arranged such that the diagonal entry corresponds to the response of each neuron to its own MEI. So here, if I just take away the highlights, hopefully you can appreciate that this is actually strikingly diagonal matrix. So not only do we observe the cell intensifier more strongly or most strongly to its own MEI, something we actually set out to show, but there are also gives very little activation to MEIs of other neurons, thus showing high degree of specificity to its own MEI. So this sounds all great. This matches our prediction of like MEI being the most exciting input, but we're very suspicious and very cautious about this. So we really wonder, well, are we really sure this most MEIs we generated are the most exciting inputs? We wanted to compare against all the other alternatives that we could think of that could be reasonable candidates for being a good stimulus to drive the target neurons. So we compare MEIs against various control stimulus. We started with the linear receptive field, which again is the best stimulus if the linear nonlinear model is an accurate model of the neuron, to also best Gabor patch that we could find from the model neurons. This will actually kind of correspond to the Osals and Field's popular normative theory of sparse coding of natural scenes in early sensory areas. We also looked for best natural image patch among 5,000 natural images that has been never shown to the model during the training, cropped to match the size and location of the MEIs, as well as the corresponding unmasked version of these natural images. And here are example 9 cells in the center. So you can maybe see a bit of a heartbeat at the very center corresponding to responses of these neurons to their receptive fields. And I was really excited to see that when I actually compare them against the responses to the MEIs, there's obviously higher activation achieved in driving these 9 cells. Like seeing something of this clear, stark difference was quite shocking to me. And this pattern actually continued and across all control images as represented. So here across the 4 different controls we tried, the y-axis always shows the activation of the target, activation against the MEIs, where each dot corresponds to a particular neuron, and axis corresponds to activation against different control images ranging from receptive field Gabor to natural masked and unmasked natural images. And across the board, MEI has been the most effective in driving the target neurons than any of the control images. So with that, I'd like to summarize the part 1 of my talk, and where I think hopefully I set out to show and convince you that MEIs are indeed the most exciting inputs for the neurons. An inception loop has now been established as a new paradigm to drive new predictions from deep neural network and to be able to test these predictions in vivo. That's the predictions arriving from the neural networks. And utilizing this, we have actually shown that the data-driven deep neural networks are actually viable targets of study to gain testable insights into real neurons. Now, here I'd like to briefly touch upon some of the ongoing extension to inception loops work as well as system identification in general. And when I talk about MEIs, I typically like to say it's just like a tip of an iceberg, quite literally so because MEI only characterizes the peak location of the complex activation surface captured by the trained deep neural networks. A natural extension would be to explore around the MEIs to gain more insights in the nature of the model neuronal responses. And one such candidate would be, for example, to explore the images that lie along contours of the activation surface in hope of characterizing, for example, the invariance properties of these neurons. I'm also working on extending this MEI generation of an inception loop approach in general to dynamic responses of neurons to movies, and therefore generating for the most exciting movies, or we call them dynamic MEIs, in hope of helping us to characterize dynamic response of neurons. And I'm realizing that these are not moving, so I think that's a glitch on my side. You just have to be convinced that this would have been like vibrantly moving movies and not so much supposed to be static. So hopefully in the future you get to see the moving versions of these. And also, more as a general point in system identification, I've been really interested in obtaining not just a good fit to the responses, but also fitting a model that will allow us to identify some interesting properties of the model neurons. And one such attempt is to build a deep neural network architecture that can discover and identify what we call functional cell types. So here we define functional cell type to be a group of neurons with shared nonlinear functions or nonlinear computation, but perhaps applied with neuron-specific transformation to the input image, such that they do same computation, but looking at the different place within the image, at the different scale and different rotation potentially. So given this, we design a network architecture that can learn these neuron-specific image transformations, as well as cell type-specific nonlinear computation, and to also learn to assign each neuron into a cell type, one of these cell types identified, and do so all together in an end-to-end fashion based on responses of large population of neurons to natural images. And in our preliminary results, we observed that this network can fit mouse V1 responses with the close to state-of-the-art predictive performances, while identifying numerous clusters of which are hypothesized to be functional cell types. So in future experiments, I'll be very interested to experimentally verify these different functional cell type clusters and to relate them to known biological cell properties, such as a cellular morphology, or to also connectomics motif. And here, obviously purely speculating, but it may be that each such functional cell type corresponds to some difference in morphology, such that the different nonlinear computation can be related to the different extents of the dendritic arbors in a way they actually collect information in a very nonlinear fashion. So now we have seen the story of how visual stimulus can give rise to population responses on the encoding front. Let's take a look at how the rest of the brain may now work with such visual information as encoded by visual population response to arrive at decisions and behavior. So recall from earlier that when presented with a cucumber, the cat jumped up. But after all, why did the cat jump up? Is this a natural reaction to cucumber? Well, at least it seems to happen fairly common behavior that is not really just specific to the one cat I happen to find on the internet. And popular explanation for this goes that perhaps cucumber, and even for human, maybe if you stare long enough or look at some variants of it, they actually look like snake. So maybe what's happening is they're misidentifying the cucumber for snake. But it turns out that they're not always, when they're not caught by surprise, they don't necessarily always jump up to a cucumber. So I believe that the key difference here between jumping up and not jumping up to cucumber lies in uncertainty in the visual observation. And let me show what I mean by that. So when the cat is presented with the retinal image of a stationary cucumber, the cat could be asking which of the two possibilities, among others, is a cucumber or is a snake? That could have given rise to the retinal image. And while a cucumber sitting stationary mostly looks like, well, a cucumber, so we can expect the cat will conclude that image is mostly and pretty much only consistent with the cucumber alone, and there's therefore low uncertainty in this observation. However, when the image becomes blurred due to peripheral vision, and also because of lack of time to accumulate evidence, the image can all of a sudden be quite consistent with both cucumber and snake. And while the cucumber might still be the best guess, if you want to conclude which is the better guess, there's much higher uncertainty associated with this visual observation. And I propose it's a difference in this uncertainty that can be leading to differences in the decisions and subsequently the behavior, whether to jump or not jump. In fact, series of experiments on multisensory integration has revealed that we, human, and also monkey, weigh different sources of information according to their reliability, which is the inverse of uncertainty, and do so on a trial-by-trial basis. And these results really provide for strong evidences that the brain encode and utilize uncertainty information in arriving at the decision, but how exactly these uncertainties represented by the brain, particularly population of neurons, has been really poorly understood. Now, a critical observation in the brain is that the neural population response is quite noisy and thus varies significantly even on repeated presentation of an identical stimulus, such as that any one pattern can actually be consistent with multiple different values of stimulus, here as illustrated by a simulated population responding to an oriented stimulus of -15 degrees. Now, fortunately, at high contrast and thus low visual uncertainty, neurons selected for the particular stimulus tend to fire vigorously, and making it relatively straightforward to predict what stimulus could have given rise to that stereotypical pattern that you're observing. However, at the lower contrast and therefore higher or greater visual uncertainty, neurons tend to fire much less, such that the resulting population pattern may be consistent with the broader range of stimulus, making it harder to discern what could have been the causing stimulus. In fact, we can formalize this notion by computing something called the likelihood function from the population response, which is the probability of observed neuronal population response pattern given any hypothesized value of the stimulus. By utilizing knowledge of how cell tends to fire to a stimulus, we can compute the likelihood function for each observed population response. And note that due to random fluctuation of the population activity, this decoded likelihood function can still fluctuate with repeated presentation of an identical stimulus. Again, at high contrast and therefore low sensory uncertainty, decoded likelihood tends to be narrow and centered close to the actual stimulus. But as soon as you go to lower contrast, and thus higher sensory uncertainty, decoded likelihood function tends to be wider, and its peak location can be quite deviated from the true stimulus value. So overall, likelihood function can capture both the best stimulus estimate through the location of its peak and the associated uncertainty via the shape of the likelihood function, namely, for example, the width of the likelihood function, where the more uncertainty there is, the wider the likelihood function tends to be. And it is precisely with this notion of the likelihood function that theory probabilistic population code is formulated. Under the theory of probabilistic population code, or PPC, a population of sensory neurons simultaneously captures the both best stimulus estimate and the associated uncertainty by representing precisely these likelihood functions, which can then be decoded or utilized by the subsequent brain areas in arriving at decisions. So it's specifically under this hypothesis, the uncertainty information is captured by the shape of the likelihood function, which again could vary from trial to trial. In an alternative hypothesis, V1 population only encodes the best estimate of the stimulus, and any uncertainty information is captured through independent path and only combined by downstream areas to finally arrive at this total information of the best guess and associated uncertainty. So again, in this one here, the difference is that the V1 population would only encode for the best point estimate. So the critical prediction is that it's only under the PPC that the trial-by-trial fluctuation in the shape of the decoded likelihood function will actually carry uncertainty information as utilized by the subject, and therefore it's only if PPC is true that we will expect that using this uncertainty information as the form of likelihood function width will be helpful in predicting monkeys' trial-by-trial decision. So to test the PPC against the alternative hypothesis, it is critical that 3 criteria are met. First of all, we need a task that requires the use of trial-by-trial sensory uncertainty to be able to tease apart the alternative hypothesis on how uncertainty is encoded. After all, if the task doesn't require uncertainty, whatever the hypothesized form of uncertainty encoding, they wouldn't be reflected in the subject's behavior. Also, given this is a population coding, naturally we need to be recording simultaneously from populations of neurons. And we also subsequently need a good way to be able to decode likelihood function from such population. And I will touch more about the challenges that usually are present in doing this procedure. Unfortunately, meeting all these criteria has been a major challenge. In fact, that there has been no study previously that has been able to meet all these 3 criteria simultaneously, and therefore has been limiting any sort of electrophysiological evidence in support of or in disapproval of theory of PPC. So given this, my approach was to exactly meet these 3 criteria. So I first trained macaque monkeys on a task where the optimal performance requires the trial-by-trial use of uncertainty. And as the monkey performed the task, we'll record from population of V1 neurons. And we actually devised a new method to decode the likelihood function over stimulus on each trial. And utilizing this decoder likelihood function, we predict the monkey's trial-by-trial decision using the models with or without the uncertainty in the form of the shape of the likelihood function. Again, if under, for example, PPC, if the shape of the likelihood function is what carries the uncertainty, we'd expect the model utilizing that to be a better predictor of the monkey's trial-by-trial decision. So again, step one lies in training monkey on a task that requires the use of sensory uncertainty on a trial-by-trial basis. And here we use a task where the objective is to classify and orient the stimulus into one of the two categories of learned classes. And this task was specifically designed so optimal performance requires the use of trial-by-trial sensory uncertainty information. And here's how a trial will go. First, a class is chosen at random between C equals 1 and C equals 2. And each of these classes define a distribution over stimulus orientation from which the oriented stimulus will be drawn. C equals 1 defines a narrow distribution over a narrow Gaussian distribution over orientations, and C equals 2 a wider distribution, Gaussian distribution over orientation, but with identical mean. We then present an orientation drawn from selected classes distribution without indicating which class it's been drawn from. And with training, monkey learns these two classes of distribution so well that they can learn to classify an orientation as either being coming from C equals 1 distribution or C equals 2 distribution. Critically, we vary the contrast of the stimulus from trial to trial, and I'll get back to this point very shortly. So if you're like the monkey and you want to perform optimally on the task, and if you consider a case of no sensory noise, well, one simply has to determine the optimal decision boundary, which actually so happens where the two curves meet. So you would just decide C equals 1 in this red region and C equals 2 for orientation falling outside of the red region, and you would be optimal at performing this task. However, the actual observations are much noisier and more difficult, such that even to a present stimulus, your actual observation is a slightly noisier version of it, fluctuating around the actual presented orientation, and this becomes particularly worse and wider at the lower contrast. As a result, because of this noise and observed orientation, the distribution of observed orientations under the two classes becomes wider at the lower contrast, and also this causes the decision boundary, which is the meeting point of the two curves, to become further apart at the lower contrast. And because I vary the contrast from every trial to trial, if this monkey was to keep on performing optimally on the task, the subject must use their uncertainty information to adjust the decision boundary according to their estimated level of uncertainty. And this is the basis for saying that this task requires the use of uncertainty to be able to perform optimally. So once the monkey has been successfully trained and learned the task, it's time to obtain the population recording from V1. We've implanted 10 by 10 multi-electrode UTArray in parafoveal V1, allowing us to obtain up to 96 multi-units recording simultaneously. We then collapse this over the 500 milliseconds of stimulus presentation duration to arrive at a vector of spike counts over this 96 multi-units, thereby giving to R, which is now used in the subsequent step of decoding likelihood functions from each trial. So traditional methods make strong parametric assumptions about how stimulus tends to give rise to responses, known as the generative model of the responses. Common choice usually includes estimating the tuning curves, which is again the expected response condition on the stimulus, and combine this with a simple noise model such as independent Poisson. While such assumption actually allows you to compute likelihood function rather easily, using wrong assumption can lead to biases in the decoded likelihood function and subsequently in the biases in the results of the study. So rather than employing strong assumptions about the generative model with possibility of biasing my outcomes, I sought to develop a novel deep neural network-based methods to directly learn to decode likelihood function from population responses. At least I set out to see, is this possible? And it turns out that this actually is rather straightforwardly possible, and here's a quick math trick that lies behind it. So if you first of all assume that there is such a network that can approximate here a discretized likelihood function over stimulus from the population response, as shown here, because as an experimenter who designed the stimulus, I actually know the prior distribution over the stimulus. Therefore, I can combine these two to come up with estimates of the posterior over stimulus condition now on responses. And it's well established in deep neural network training on classification tasks that such posterior estimate can be trained to approach the true posterior through the use of cross-entropy loss against the actual stimulus S that was presented to obtain the response in the first place. So hence, by training this to achieve through the cross-entropy, the true posterior, we can backwardly make the output of the network achieve the likelihood function up to a multiplicative constant, which suffices for our very usage. So my very last step lies in comparing models to predict monkeys' trial-by-trial decisions. And under the full likelihood model that corresponds to probabilistic population code theory, again, the likelihood function is decoded from trial by trial using the very deep neural network we just discussed from the V1 population. And under this model, both the center and the shape of the likelihood function would change from trial to trial. In the alternative non-PPC model, V1 population only provides the best estimate of the stimulus, and thus uncertainty information is again not represented in the shape of the likelihood function, even though you still could decode this from V1. Given this, when you actually consider trials from the same contrast, there's actually no reason to expect that the uncertainty information should change from one trial to another, and therefore this gives rise to the fixed uncertainty model when conditioned on particular contrasts we present. And here I've just very briefly discussed, again, in the formulation of the fixed uncertainty model, we also utilize a deep neural network, which essentially learns two things. It learns a fixed shape per contrast of the likelihood function. So this corresponds to a fixed degree of uncertainty shared across all trials from the same contrast, and a network that actually learns to take the neuronal responses into a value that will learn to shift this shape from left and right across the orientation space, such that, again, this corresponds to that across the trials from the contrast, only the center of the likelihood function will shift without changing the shape of the likelihood function. So to make this point even further clear, let's consider trials from the same contrast under both models. Under full likelihood model, we expect both the center and shape of the likelihood function to change from trial to trial. Whereas in the fixed uncertainty model, again, only the best point estimate is extracted from the trial by trial changes in V1 population responses, while a fixed shape of uncertainty is utilized, therefore reflecting the fixed contrast level. We then take these two models of uncertainty encoding and feed them through a Bayesian decision-making model, which utilizes knowledge of the task and prior over classes to compute the posterior over stimulus classes, which in turn yields the trial-by-trial predictions of the monkey's choices. And again, critically, if the trial-by-trial fluctuations in the shape of likelihood function that you can decode from V1 population is actually what carries the uncertainty information that is used in the decision-making process, we will expect the full likelihood model that carries exactly this information to be able to perform better in predicting monkeys' trial-by-trial decisions. So without further ado, let's just dive into the results. In comparing the two models, I plotted the difference in the performance, taken as the difference in log likelihood of predicted performance, where the fixed uncertainty would be the 0 line. And across both monkeys and across a wide range of contrasts, the full likelihood model successfully yields better trial-by-trial predictions for both monkeys' decisions. Therefore, this in turn suggests that the trial-by-trial fluctuations in the shape of the likelihood function that's carried by the full likelihood model is indeed informative about monkeys' decisions. So in summary, again, I just shown that the trial-by-trial fluctuation in the shape of the likelihood function indeed appears to be informative in predicting monkeys' decision. And this in turn provides for an evidence, and in fact, the first population-level electrophysiological evidence in support of the hypothesis that population of V1 neuron encodes uncertainty in the form of the likelihood function on trial-by-trial basis, thereby giving support to the theory of the probabilistic population code. Again, the last stretch of a minute, I would like to briefly discuss some potential extension that's going on at the moment. So now we established that the V1 potentially represents information in the form of likelihood functions. As an ongoing work, I'm interested to know how is this likelihood function from V1 propagated to other brain areas. In particular, I'm working on asking how this information on V1 propagates to categorical decision-making areas such as prefrontal cortex. To assess this, I'll be recording from V1 and PFC simultaneously, and decode likelihood function over classes based on each population responses separately. And in particular, I'll be looking for signatures of the likelihood function as decoded from the the PFC and see how it relates or how it could be predicted from fluctuations in the likelihood functions as decodable from the likelihood functions as from the V1 populations. All right, so I guess just to quickly conclude, as an overall summary, in the part 1 on encoding, I presented deep neural network-based models that offers the state-of-the-art predictive performance, and utilize this in a novel closed-loop paradigm we call the inception loop to provide us with the methods of taking predictions from such network and turn it into experimentally testable predictions. And we have successfully applied this paradigm to derive the most exciting inputs for the neurons. In the part 2 on the decoding, we provided for the first population level electrophysiological evidence in support of theory of probabilistic population code through carefully designed task and analysis. And particularly the exciting part I found was that we made use of deep neural network-based methods to be able to decode likelihood function on trial-by-trial basis, doing away with the common and problematic assumptions that was common in being able to decode likelihood functions from populations of neurons. So with all of that, I think I've been a couple minutes over, but obviously no such work can be done without having a great set of collaborators. And I'd like to thank specifically the SENS lab, my postdoctoral lab at the moment, as well as my ongoing continued collaborations from my advising labs of Andreas Tolli's lab, Weijie Mao lab, and my experimental collaborator, Hendrik Nienborg and NIH, and as well as my brain tank collaborator in the sense of thinking through stuff with Alex Ector at University of Göttingen. And with that, I'd like to open up for questions. Thank you.